you know, as long as you kind of stopped anyway, what? All right. So, questions about the exam? No surprises? I mean, and the average score was 89, so it's not, so a little easier maybe than the second exam. I don't know. Or, you know, or you're getting better, or my teaching's getting better, or whatever. I'll take whatever we can get. So you get the idea. And any questions? Yeah, good. Angelica.
Kind of just really have the second epilogue.
Will
the final be 8-9 in the epilogue?
The final will be comprehensive, so it will be over everything, but it will certainly emphasize 8-9 in the epilogue. And yeah, good.
And it's all good up our exact method.
What about the final?
D12, it says the final is out of 96 points. Is that true?
No, it's not true. I don't. D2L has so many places where you. So there was a time there were only 92 questions there, and I guess that hasn't gotten it. But I assure you, there's 100 now.
Is there a possibility you can write some extra credits for you?
And several people have asked about extra credit, and no, I don't want to do that. For one thing, you know, my lawn doesn't need mowing anymore, and so it's like, yeah, in general, I try not to do that. If folks have had problems with Kahoot or the Perusall for some reason, if there's a reason why they're not making that, then I want to hear about it. But otherwise, and as far as I know, actually I haven't looked lately. There were a few folks that looked like they might get in trouble with getting enough Perusall points. And Grace pointed out to me that, you know, in the syllabus, I said, well, there are 10 readings, and so you know, or 9 readings, and 27 you could have a much of 27, so you got to get 20. And she pointed out to me that for some reason the interlude, I meant for it to be lumped in together with chapter 8. So now you've got another opportunity to get 3 points from the interlude that's separate. Didn't mean to do that. And of course, I added a chapter. So now, even if you discount the first one, remember, you don't count the first one because that was for everybody to figure it out. Even if you do that, there's like plenty of opportunities to make the, and thanks to Grace, I just looked at it, the threshold of 20 points of Perusall. Yes?
At least they said they did kind of like things off to support again in fixed result.
The exam is based on the 4 exams. Perusall, as long as you make the threshold, it does nothing. If you don't make the threshold, then your grade gets reduced 1 notch. Likewise, if you don't make whatever I put as the threshold for Kahoot's participation, okay? Because I want you to participate. No, I do have a slide for that. Good. Abner.
The final the same weight?
The final will be the same weight. Oh, another thing is officially final exams, you're supposed to get 2 hours, and so you get 2 hours. But it's the same number of question. And so, there. Okay. We've come a long way. So, we have had a fertilized egg that divided plenty of times and eventually started making a nervous system. And we've concerned ourselves primarily with those organisms like ourselves where the construction of the nervous system is depending on cell-cell interactions. But of course, the cell-cell interactions are those that are already programmed into the cells based on the genome that they received, right? And eventually there's a neural tube. I'm going to emphasize vertebrates. Eventually there's a neural tube, lots of division going on, neurons get made, they migrate out to various places and populate the brain, a lot more at the rostral end of the neuraxis than the spinal cord, but still billions of neurons. They, having gotten to the right place following adhesive cues and diffusible cues and things like that from other cells. They have started to differentiate into neurons, and first thing they do is send out an axon, and the axon has had to feel its way through the body and the nervous system to get to the right target. And what happens then is the synapses mature. I think I'm at chapter 6 now. And the synapses mature, and eventually some of the synapses, some of the neurons don't get enough trophic factors and from their target and therefore die, but everything's fine because that's all part of the plan. And then there are also long distance messengers going on to the chapter 7.5, where some parts of the body exert control over other parts of the body, including the brain, by secreting hormones. And the hormones act on the cells to do various things. And they will, for example, push an insect into the stages of metamorphosis, or they will push frogs into the state of adulthood. And thyroid hormones in other vertebrates is absolutely crucial for make sure you make enough neurons in enough places and that they work properly. And for those species in which there are two sexes, in vertebrates at least, it's going to be gonadal hormones that are going to send signals throughout the brain and the body and have the brain take on a phenotype that's appropriate for reproducing either as a male or a female. In all of this, no outside entities had a say in what happened next, right? What I just described, all of that could have taken place inside a sealed capsule on the space station, right? And everything would have happened. Well, okay, I forgot about gravity, but you put it, you spin it so you have the right amount of gravity, and it would happen there. And there's been no outside information that's come in. And that, to my mind at least, sounds an awful lot like a Platonic or Cartesian soul, right? Where there's a whole bunch of information that got put in there and nobody else had a say in it, and it had nothing to do with what's going on in the world that's spinning around with us on it, right? But now we reach a stage where natural selection hit upon another way to provide more information into this organism. Note that up to this point, the only information that's gotten in there was the information that was in the genes that it inherited. And you could make a pretty complex nervous system that way. That's where we are right now. It's pretty complicated. But in the outside world, it would have no chance. Never reproduce, right? It's not nearly enough information if you're going to make really complicated organisms. I mean, if you're going to be C. elegans, okay, you can probably get by with that. But for any more complicated organism, the only way it's going to have a chance to reproduce is if natural selection can somehow find some way to shove more information in there. And what's pertinent information that you want the nervous system to know? What's going on in the world? What's it like out there? How long's the day period, right? What are the temperatures like? Where's the food? I mean, there's so many things that if you can get that information into the organism, you're going to make it more fit. It'll be more adaptive. And so long ago, natural selection found another source of information beyond that of the genome, and that was neural activity. And so what we're going to talk about today are those cases where the decision about which synapses come and which synapses go is not dictated in the genome. Rather, it is being determined by neural activity, whether an action potential happens or not. And to some extent, we're going to talk first about spontaneous activity. Turns out once you make a neuron, once it's got all those, expressing all those genes to be a neuron, it's going to fire action potentials once in a while, right? There's so-called spontaneous activity. You can't be a well-differentiated neuron and not send an action potential once in a while. So what we're first going to talk about are those cases where the action potential is not triggered by outside events. But the reason I'm spending time talking about that is because the rules for how spontaneous activity affects synaptic fate are basically the same for the rules about how experience affects synaptic fate. And of course, the one led to the other, right? There would have been no way for experience to affect synaptic fate unless neural activity affected synaptic fate. How are you doing? And so first we're going to talk about spontaneous activity, which was the old chapter 8. And then we're going to talk about experience-driven synaptic activity, where sensory axons are detecting what's happening in the world and using that to decide which synapses come and go. And then the last chapter will be how once sensory systems are online, then it's possible to shove more information into an individual's brain by having conspecifics, other individuals of the same species, having them impart their wisdom or mistakes into the brain. And by the time that's all over, you got, you. Okay? Okay. So, oops, wrong way. Oh, I meant to, yep, but you had it in your notes. One place where synaptic activity plays a role harkens back to apoptosis. The hallmark example of apoptosis, where the individuals make more neurons than they're going to need, is in the creation of spinal motor neurons, where normally somewhere between the 5th day of incubation and the 13th day of incubation, half the motor neurons that got made get lost. Not because of a catastrophe, not because they weren't, perfectly valid neurons, but because that way you end up with a matching of the number of motor neurons the size of periphery. But in fact, people were surprised when they learned that apoptosis is itself dependent on neuromuscular activity. If you use drugs to block neuromuscular junctions at that critical stage, the motor neurons just sit there. They don't die. They just sit there. And so you can go till hatching and have an individual chick with twice as many motor neurons as other chicks. But as soon as you lift the blockade at the neuromuscular junction, then cell death will happen, and it will be about the same amount of cell death as would have happened if you hadn't stayed out of the way in the first place. So there's one case where neural activity, not sensory-driven neural activity, but where neural activity is playing a role in apoptosis. Questions about that? Okay. But I promise you we'd talk mostly about synaptic fate. So what happens next after making a surfeit of neurons, half of which die in various parts of the nervous system, what happens next is the nervous system makes a surplus of synapses, where you have an individual that has more synapses in their brain than they're going to need and more than would help them function optimally. And for a long time in the field, this period was known as synapse elimination, because what really struck people, just as it struck people that neurons that had been made seemed to die pointlessly, it struck people that synapses had been made seemed to be disappearing pointlessly. But eventually people realized that at the same time that synapses were going away, and that's what we'll talk about first, that other synapses were getting made. And so that led to, and I would say this is about 1990 or so, where a greater realization that the nervous system is much more plastic than people had realized before. And that'll be a theme that we'll see more and more often here. Okay, so eventually then, when they realized that synapses were coming and going, and there might be a net loss of synapses, but still there are so many more synapses getting made, et cetera, it made more sense to call it synapse rearrangement rather than synapse elimination. But you'll see the other phrase in the field. And as in almost everything in the neurosciences, most of what our best insight into a process is at the neuromuscular junctions. And for the same old reasons. Here's this great big synapse out in the periphery. You don't have to do much surgery to see it. and you don't have to do much surgery to monitor the targets. And so in motor neurons, the way your nervous system is organized, the way your muscles are organized, is in every muscle you have thousands of muscle fibers, and each individual muscle fiber is being innervated by one and only one motor neuron. Okay? But there's no situation where it's one motor neuron to one muscle fiber, right? Rather, and by the way, contracting one of your muscle fibers, you'd never even notice it. There'd be no noticeable change in the tension of the muscle, right? Rather, each motor neuron is innervating many muscle fibers, typically hundreds, sometimes thousands. And every time the motor neuron fires, the action potential goes down all its branches. It goes down one axon to the muscle, and at the muscle, it branches, typically at the very last moment, and then innervates several different muscle fibers. And it turns out neuromuscular junctions in adult animals have a very high safety factor. That is to say, virtually every time the action potential reaches the presynaptic terminal, it will fire, and it will cause a contraction in the muscle fiber. And so this is how you control your muscle. And when you want to move a muscle a little bit, you only turn on a few motor neurons, and each one of them acts on its posse of muscle fibers, and they contract all at the same time. We call that motor neuron and the muscle fibers it innervates a motor unit. And you control the extent of contraction of a muscle by recruiting more and more motor units so that you get more and more contraction of the muscle. So far, so good. And hopefully you heard some of this before, somewhere. Nod or shake your head. Yeah. Okay, good, excellent, but worth remembering. And it turns out that when you were younger, now let's go to rats. In rats, at least, motor units are organized the same way they are in you, but in adult rats. But newborn rats, when people were looking at neuromuscular junctions, they were quite shocked to see that during development, every muscle fiber is innervated by several motor neurons. And what's going to happen is, eventually, over time, each muscle fiber will end up with only one motor neuron pre-terminal, even though it had several before. So many of these terminals will retract, leaving each muscle fiber with one and only one motor neuron innervating it. And once that decision gets made, the other thing that happens is the remaining motor neuron starts making that neuromuscular junction huge, right? Filled with millions of acetylcholine receptors, lots of vesicles that are get released each time, because again, you want to have a high safety factor at a neuromuscular junction, right? If you need to run, you really need the motor unit to cause those muscles to contract. In some ways, notice that this is a net gain in synaptic strength, because these little synapses in the newborn rat, they're kind of puny. And when each one of them fires, it won't necessarily cause the muscle fiber to contract, as opposed to once you get into adulthood. In fact, if we just measure how big is the neuromuscular junction here and the sum of the neuromuscular junctions there, there's no comparison. These are huge. So there's a net increase in synaptivity, if you will. And I want you to understand how we learned about this. Oh, and the other term to use is, at this point, if each fiber is being innervated by more than one motor neuron, we say it's polyneuronal innervation, right? That'll be useful terminology. And here's how it was determined, which is take a newborn rat and dissect out a motor nerve and stick an electrode in a muscle fiber. You stick something in the bath with the muscle fiber there so it never contracts and pushes out your electrode. But now you can record all the EPSBs that arrive from axons. And so you stick an electrode in a given muscle fiber, you you put an electrode on the nerve, the motor neuron stimulating it, and you give a very low dose of electricity, a very, very low pulse. And at first you get no EPSPs. Everyone get, because you didn't give a big enough stimulation of the nerve to get an axon to send an action potential. But as you increase the strength of stimulation, eventually you will activate one of the axons that's innervating your particular muscle fiber, and you'll measure the EPSP. Simple. But what they found is, if they increase the electrical stimulation a little bit more, sometimes they got a bigger EPSP. Oh, and I guess I should have said, in this sort of situation, when it's a low stimulation, every EPSP looks the same. It's always the same size, always the same latency, always the same length of time that it's there. And that makes sense because you've just activated one synapse. And the way synapses work is when they release vesicles, they try to release the same amount of vesicles every time. So of course you'd expect to see a same size EPSP. But then they noticed if they increased in newborns, sometimes they'd get a bigger EPSP. And indeed, you could plot out the sizes of the EPSPs, and it was totally binary, right? Either they got a little EPSP or a twice as big EPSP or nothing in between. And this was interpreted as the increased stimulation had recruited another axon. And you increase the stimulation some more, and now you get a complicated EPSP where it's bigger, right? But also you can see that there's a later arriving EPSP. And so in this case, they knew that this muscle fiber had at least 3 inputs. 3 different axons were innervating it. Everyone get the logic of that? And notice that that's a conservative number. It's always possible there's a 4th axon in there and your electrode just isn't reaching it to stimulate it. But at the very least, there are 3 inputs to this muscle fiber. Yeah, after.
There's two with the stable axon that we got.
I hope I didn't misspeak. I mean, in this case, when they see 3, they think it's 3 different axons.
Yeah, but why wouldn't they assume what's evidence that it's not just one axon, so it's 3 things?
Well, I mean, so yes, you'll be. One of the things that's going to change is everything's different once you look at adults, right? But also, that is just, and that got worked out in the 30s and 40s. That's the nature of axons at neuromuscular junctions. Is each time an action potential comes down, it releases one particular batch of vesicles, and therefore you get one particular sized EPSP. And they're very regular. Yeah. And that's the other thing that tells you. I mean, if in fact it's one axon that's doing all these things, well then why wouldn't there be a continuum of EPSP sizes, right? You wouldn't expect them to be in increments. Okay. Anyway, then you get other rats and you keep doing these sorts of things, and you see that in the first week of life, every muscle fiber, 100% of them are polyneuronally innervated. And as time goes on, by the time they're weaned, absolutely no muscle fibers are polyneuronally innervated. Each one is innervated by one and only one input by this method. And here's the same data, just plotted differently. And what I want you to pay attention to is how much variance there is. Look how huge the standard error bars are. And by the time you get to 2 weeks of age, there's no variance. Every darn muscle fiber you find has exactly one input. Importantly, not only are there no muscle fibers that are getting no input, that are getting more than one input, you don't see any muscle fibers that are getting no input. And that tells you something important about what's going on here. Because if we go back to this cartoon, if you got rid of polyneuronal innervation because every muscle fiber decided, you know what, I'm going to retract 60% of my branches. Well, right? And if they all decide that, that would also mean there'd be a loss of polyneuronal innervation, right? But notice if it was being done that way, then there'd be some muscle fibers that would have all of their axons retracted. And you'd expect there'd be at least some muscle fibers where 2 were still left. Rather, the fact that every muscle fiber ends up with one and only one input, that can only happen if there's some sort of competition going on between the inputs, or at the very least, cooperation or communication between the presynaptic fiber and the postsynaptic target. And that's the theme of this course, right? Development proceeds because of cell-cell interactions. And here we can see pictures of that in action. This is a preparation where different motor neurons have different colored fluorescent molecules in them. And so here's a postnatal day 11 junction, and they're huge, where there's a green input and a blue input. The red is just a marker for the acetylcholine receptors. And 2 days later, on P11, it looks like the blue motor neuron is predominating. 2 days later, and so they went back, put the animal back in its cage, came back and looked at the exact same neuromuscular junction. Tough work to do. 2 days later, it looks like the green's going to take over. 2 days after that, you can actually see what's called a retraction bulb, the end of the axon, of the blue axon, coming away. And 3 days after, the day after that, here's a fiber that's being innervated by only one input, the green motor neuron. And notice there's a complete overlap that the presynaptic terminal is only on those places in the muscle fiber where the acetylcholine receptors are. There's a perfect match, 1-to-1 going on there. Good. Or
Like that big maybe day.
how can something like that be done with the animal still alive? It's tough work, right? So, and one of the things you probably didn't realize, yes, you have to take care of the animal, make sure they're still going to be okay. The thing you might not realize is you got to find that same synapse, that same, again, this is the sort of thing that's a lot easier to do in a peripheral muscle than in the nervous system, but you better have a pretty good idea of where that neuromuscular junction was and come back and find it. Yeah, yeah. Well, and I didn't participate in this one, but I've done this kind of research, and I can tell you the other thing is each time you look in the animal, you try to record as many neuromuscular junctions as you can find, because you don't know which ones you're going to be able to find next time, right? So it's also a bit of a numbers game. And the animal's alive, and they're on the microscope slide, on the microscope stage, and you put a cover slip over the muscle, and you're looking through the, trying to find where was that junction, right?
I'm going to stay through.
As they stay still. I'm not going to ask any more questions. Okay, now I'm going to lose my audience. They stay still because you've given them curare and they're on a ventilator so that they stay alive. They're also anesthetized, but that's, yeah. And the biggest movement artifact you have to deal with is heartbeat, because you want them to stay alive. Okay. Anyway, this sort of synapse elimination is probably happening all over. Another place where it got well documented was in the cerebellum. So I have to tell you a little bit about neuroanatomy, or the circuitry of the cerebellum, that you might not remember if it ever came up earlier, which is that those Purkinje cells that we saw earlier, those giant cells, they are being innervated by lots of different targets. But one of the places they're being innervated is by brainstem neurons in the inferior olive that send their axons up to innervate the Purkinje cell. And again, in an adult, every Purkinje cell receives one and only one input from a inferior olive cell. But if you look earlier in development, Oh, and those inputs from olivary cells to the Purkinje are known as climbing fibers. You'll see why in a moment. So they did exactly the same sort of experiment. This time they're sticking an electrode in the brainstem, not on the muscle nerve. And you can see that if you increase amplitude, first you see one EPSP that's at this time point, and pretty soon you see, if you keep increasing the strength of the electrode, you'll see more at that point, and then you'll start seeing various bumps, etc. And these are averages of many traces. And so what you see is that there's always a discrete number of EPSPs that you're recording. And while on the 5th day of life, every Purkinje cell is getting input from at least 3, an average of 3 climbing fibers. By the time you get to 2 weeks, every Purkinje cell is getting input from 1 and only 1 climbing fiber. So those other inputs have been eliminated. And just as with the neuromuscular junction, the other competitors go away and the remaining axon makes a really big synapse. So too in the cerebellum, where the remaining climbing fiber, which has originally been only on the cell body of the Purkinje cell, by day 15, where each climbing fiber has exclusive access to that Purkinje cell, then the synapse starts migrating so that eventually the entire huge Purkinje dendritic tree will have thousands of synapses, all coming from that one olivary cell in the brainstem. So there's been an elimination of inputs, and at the same time that there's been an elimination of inputs, there's been this vast growing of synapses.
Yeah, that's
Good.
There beneath the class, I asked a question about Purkinje, though you said it's like an onion sheet. Can you send me it's pretty that way?
Oh, I didn't mean to say I would explain that later. After class maybe, but no, it's remember the granule cells that parachuted down through the Purkinjes, left their neurite behind them, trail, and those axon, the branch was going perpendicular to all the Purkinje cells, etc. So those granule cells, every time that granule cell fires, its action potential goes up here and hits this Purkinje cell now, and the next one, and the next one, and the next one, and the next one. Seems like a timing device. And we know the cerebellum is crucial for timing, especially in motor planning and stuff. Yeah.
So could one islet cell innervate multiple pathology cells?
Great question. They do, right? So I'm emphasizing the synapse we can see, but what this means is at first every olivary cell was innervating several Purkinje cells, but as the process goes on, each one ends up with only one target, and each olivary cell ends up with only one Purkinje cell target. Whereas before they were innervating several.
Yeah.
And then the other thing you want to know, and we've got no handle on this, is, well, so what's happening to the inputs to the olivary cell? So that now I can make that Purkinje cell, I can stimulate that Purkinje cell and only that one out of the billions, as long as I stimulate that olivary cell. And so you figure synaptic input to that olivary cell also has to be just right. And I don't know anything about that, except I know that usually it works out.
I don't feel like how the substation needing to only the Purkinje properly to see the axon from one cell.
Okay.
What is there a mechanism so that the islet cell doesn't go to a different Purkinje cell and also innervate it?
There may be, but we've got no handle on that.
Okay.
What gave the surviving olivary cell an edge that it stayed? No idea. But it probably matters.
Baseball pool again,
And
to see the foots to be on that one can be dull, and then when it's like the motor neurons innervating also off the
right. And climbing fibers aren't like that. It's okay. And now you're making me uneasy, but I'm anyway. So let's leave the story at that, because it's time for Kahoot. A lot of learning objectives, but a lot of this is going to be repeat for you. It still strikes me as strange that a psychologist, a guy, just by thinking about how synapses and neurons work, came up with an idea about how synapses might come and go that turned out to be right in so many ways. So he had done almost no neural work himself. And in 1949, he publishes this book called The Organization of Behavior that was incredibly influential. And his name was Donald Hebb. And he was a Canadian by then. He's at McGill. And the book was a bestseller. Lay people bought it because in it, he talked about all sorts of ways in which the nervous system would mediate behavior. This at a time when we knew very little about the nervous system, really, and including how it might be possible that learning could be encoded into circuitries in the brain. And so he proposed that there might be some synapses that behave in the following fashion. And now I'm just trying to see if I included it there. Yep. Where he said that when an axon of cell A is near enough, so he said, to excite cell B and repeatedly takes part in causing it to fire, that synapse will get stronger. Compared to, so let me just, he was more succinct than I could be. When an axon of cell A is near enough to excite a cell B and repeatedly or persistently takes part in firing it, some growth process or metabolic change takes place in one or both cells such that A's efficiency as one of the cells firing B is increased. And even though he felt like these were ideas that had been out there for quite some time and he was just putting them together, he was always embarrassed that people right away started calling these Hebbian synapses. And there's several things about it I want to point out to you. One is that when he says a change takes place in either one or the other, notice that this synapse could get stronger either because the presynaptic cell is releasing more neurotransmitter or because the postsynaptic cell has put more receptors there, right? Either one of those would increase the efficiency of the synapse. And notice he was not saying that active synapses will get stronger. It's not enough that the synapse was active. It has to be that the synapse was not only active, but that the postsynaptic cell fired because of it. So it's not active synapses that are maintained. It is successful synapses that are maintained. And he perceived it as a change in the size or strength of synapses. This was at a time when the dogma was, by the time a child is 10 years old or so, the number of synapses never changes in the brain. Right. And so he didn't conceive of the idea that some synapses might come and some might go. That sort of structural change was thought to be very unlikely. So he was thinking about same number of synapses, but some synapses get stronger and some get weaker. And as you go through the book, he provides various diagrams that don't look that much different from this, except they're not in color, where he could explain how that kind of a synapse could explain classical conditioning, right? Or other kinds of learning, or habituation, or other psychological phenomena that people knew about. Today we know that a synapse can get stronger, an input can get stronger, not just by increasing the strength of a given synapse, but by increasing the number of synapses that happen. Questions about that? Note that in this formulation, he's simply talking about what can cause the synapse to get stronger. But the other side of the coin is that a cell, when it fires, rarely causes the postsynaptic cell to fire. Its synapse will get weaker. And today we know that the synapse might not just get weaker, it might get retracted altogether. So there'd be another structural change. Good. Leslie?
It's explaining that we're like the cells get firing better on this.
In his diagram, another part of the circuit, it's not just this one, right? Another part of the circuit is some part of the brain that tells it, hey, that was useful information, right? But, and that therefore the synapse stays if that happens. But if you don't get that input, so habituation, right? So you're sitting in a coffee shop and every time the door opens, you hear a ding, right? After a while, you stop responding to that. It's habituation because your ears are still working. The bell's still there, but you no longer have a response to it. So in his diagrams, there'd be some part of a circuit would have to say, nothing's changed, nothing's happened. And that input gets put into it. And I don't remember the details about how he did it. But yeah, I'll bet it's online. It's not in the public domain yet, but I'll bet you can find the whole thing online. It's very interesting. Stone.
You can have a copy of the sculpts that I just occasionally follow, please.
Tone, I'm a neuroscientist and a psychologist. Of course I have a copy of this book. I mean, I'm a little shocked that you don't have a copy, but and you can't have mine, but you can certainly borrow it. Mine even has a dust jacket filled it, right? It's torn, but and I never met the man. He passed away before I became a neuroscientist. But I've known lots of people who knew him, and they all loved him. And there are many things that he pioneered that you don't know that he pioneered it. So the idea of self-stimulation that a rat would push a bar to get electrical stimulation in part of the brain, that was his idea. And he told some folks in his lab to pursue it, and they did. And they never even put him on as an author. Sensory deprivation, sensory isolation tanks, that was Hebb's idea. Right. So, he's an active guy. Okay, enough of that. So he said that in 1949, but it would take like 30 years almost before anyone actually found a synapse that behaved in the fashion that Hebb predicted it would. And they did this looking in the hippocampus. And this is just to remind you that the hippocampal formation consists primarily of the hippocampus, which is so named because that's the Latin word for seahorse. And you can see how it's kind of shaped like a seahorse. And the hippocampal formation consists of the hippocampus proper and the various cortical regions around it. More specifically, if we take a cross section through the hippocampus, there are two groups of neurons that make up the hippocampal formation. Within the hippocampus itself, there's a line of cells that are pyramidal cells in the hippocampal formation. And then there's in the hippocampus proper. And then there's a dentate gyrus, which is part of the hippocampal formation, but not the hippocampus proper. And it consists of a sheet of granule cells. So far, so good. And the circuitry of the hippocampal formation is very well known and described and has been from anatomical preparations back in the early 20th century. Cajal loved the hippocampus, and so he mapped out all of these inputs. The structure of the hippocampus is rather complicated, and so I'm going to offer you an analogy that you can think about to see if that helps you understand it. Because notice here, we're just looking at a cross section, in this case, in a horizontal section, right? We're looking down on it there. So imagine that group of pyramidal cells in the hippocampus proper, first think of them as being in a sheet. And now bend the ends of the sheet so that it's got a U shape where the pyramidal cells are. Everyone got that? And now that's the cross section is U shape, but the whole thing kind of looks like a rain gutter.
Right.
And now imagine the granule cells in the dentate gyrus also being a sheet, and bend them down, and you've got another rain gutter shape. And now put the lip of the granule cell rain gutter into the trough of the hippocampus, the hippocampal rain gutter. Everyone got that? And you take a cross section of that, and you get something that looks like this. One upside down U and the other upside down U with its legs sticking in it. You're almost there. Now take those two rain gutters and bend them to make a C shape. Right? And they're still, in every cross section, they're still going to have that relationship between each other. And now stick it inside the brain and have the ends of the C face front. And when I say inside the brain, I mean underneath the parietal, occipital, and temporal cortex. Okay? So, and if you don't have the three-dimensional image of that, it's okay. If I could think of a way to put it on a multiple choice question, I would, but who could do that? Anyway, and then within the hippocampal formation, there are granule cells that innervate one part of the hippocampal formation, and these are known as so-called mossy fibers. And there are various collaterals, etc. I'm not going to test you on any of that, but just know they're well worked out. But then there's another pathway that Cahal loved, where there are these inputs from nearby cortex, where axons seem to be perforating, going across this is the lateral ventricle wall, perforating the hippocampal formation and going to innervate various fibers. So this is known as a perforant pathway. It's coming from what's known as entorhinal cortex that provides a major output, input. And there are three different pathways shown here in three different colors. But I'm going to tell you what people found first with the perforant pathway. Eventually, folks found that all three of these pathways behave in a Hebbian fashion. But the first demonstration was in the perforant pathway back in the maybe mid-late 70s, where they did a preparation like this, where they put electrodes into the hippocampus. Actually, at first it was in slices of the hippocampus, but whatever. They put one electrode into the postsynaptic cells, and they use another electrode to stimulate the afferents. Which afferents? So they're stimulating these afferents to provide input to these cells. Okay? And they found that if they did the stimulation like every half second or so, which in the nervous system, that's pretty slow. If they did it every half second or so, the postsynaptic potentials were very stable, stayed very much the same. And they could record these hours. And for hours, the size of the input, when they stimulated the input, the size of the EPSP, the output, was the same. And then they did something strange. They provided a tetanus, where they stimulated the axon not once every half second, but hundreds of times per second for a period of time, maybe 15 seconds. So there's just a barrage of action potentials going from the presynaptic terminal to the postsynaptic neurons. Everyone got that? And of course, when that's happening, there's no point in recording from the postsynaptic cells because the stimulation from your stimulator has totally overwhelmed your recording device. But after the tetanus is over, then you record the EPSB. And interestingly, immediately after the tetanus, there was actually a reduction in the size of the EPSB. But within a few minutes, it recovered. And that reduction was probably that you had temporarily depleted the vesicles to provide input, right? What was more interesting was that in the time that followed, in the course of many minutes, the input got stronger and stronger. And they would eventually do it in awake rabbits. And they found that every time they provided a tetanus, so here was the control pathway, very stable. But every time they provided a tetanus, the strength of the input went up. And if they provided a second tetanus, it'd go up some more. And a third, it would go up some more. And a fourth, so that eventually they had a response that was almost 3 times larger than it had been before. And we're not showing it here, but in the rabbit preparations, they found that this increased strength of the synaptic input could last for weeks. And so they named it. This increase in synaptic strength, they call it, that's a potentiation. It's not used a lot in English, but potentiation just means make stronger. What's the opposite? Attenuation, right? Nobody knows this stuff. Anyway, but so they called it potentiation, and because it lasted so long, they called it long-term potentiation, or LTP. And people were very excited about this. Number one, because it behaved in a Hebbian fashion. When they provide the tetanus, they are making that input very successful at driving the postsynaptic cell, right? Because it's getting overwhelmed with all this synaptic input all of a sudden. And later they would confirm that the postsynaptic cells were indeed being fired by the tetanus. And so the synapse had been really successful in getting postsynaptic cell to fire, and therefore it got stronger, just like Hebb said it would. And so that was one reason they were excited. The second reason they were excited, it was already clear that the hippocampus is crucial for memory and for learning and memory. And this sure looked like a memory device, right? A way to change synaptic strength permanently, or at least long term, maybe long enough to like encode a memory. How's that? And I guess the third thing was everyone was excited that this might be a way for memories to be encoded. So lots of people started studying LTP. You've heard about it. And it turns out that in those pathways, there is one particular neurotransmitter receptor that is responsible for LTP in the peripheral pathway to the hippocampus. And so first I need to tell you the input is using the neurotransmitter glutamate, which is just a small amino acid. And glutamate is almost always excitatory, and that's the case here. So the inputs are releasing glutamate every time they fire. And when we're looking before the tetanus, almost all of this EPSB is being generated by one particular type of glutamate receptor. And that glutamate receptor is known as the AMPA receptor, which we're showing here. So the presynaptic cells send their action potentials down. They release glutamate in the synapse. Glutamate acts on the AMPA receptor, and the postsynaptic cell has an EPSB, or eventually will fire. So far, so good. It turns out there are many different kinds of glutamate receptors. Some glutamate receptors that you've heard about are metabotropic. That is, when glutamate hits the receptor, it doesn't open an ion channel, but it causes changes in G proteins and stuff like that that can affect the postsynaptic cell. We aren't going to be talking about metabotropic glutamate receptors here. Other glutamate receptors are ionotropic, which means that the receptor itself includes an ion channel. And when glutamate hits the receptor, it causes the ion channel to open. And so AMPA receptors are ionotropic glutamate receptors. Oh, and why are they called AMPA? They're named that because of a particular synthetic compound that's really, really good at binding them. So I'm not going to bother telling you what it stands for because it doesn't matter. But there are other ionotropic glutamate receptors that are not AMPA receptors, but rather they're named for a different synthetic molecule that stimulates them, which is NMDA, for N-methyl D aspartate, maybe, I don't know, NMDA. And so both receptors are present in the hippocampal pathway, but when you're releasing, when the presynaptic cell is firing regularly but slowly, only the AMPA receptors are responding because it's the same glutamate that acts on the NMDA receptor, but the NMDA receptor is weird. Because for the NMDA receptor, its ionic pore is just the right size so that when glutamate hits it and the pore opens up, the pore immediately gets blocked by an ion of magnesium. And why is the magnesium wanting to come in there? Positively charged magnesium, because it's positively charged, and the interior of the cell, of course, is negatively charged, right? But because of that, no other, and the magnesium is blocked there. It doesn't go in, it just jams up the pore. Count that. So normally, at the slow pace, only the AMPA receptors are responding. The NMDA receptors are getting the glutamate too, but no ions pass. What happens that's interesting is that when there's a tetanus, and this synapse has been very, very active, hundreds of times per second for several seconds, weird things going on. One thing is because of the AMPA receptors, the postsynaptic cell is being depolarized. And when the postsynaptic cell is depolarized enough, then there reaches a point where magnesium is no longer running down its concentration, is no longer being attracted, and so the magnesium block is lifted. And when that happens, the NMDA receptor doesn't let sodium and potassium ions in to depolarize the cell further. Rather, their channel is specific to calcium, and the external calcium runs in and starts changing the synapse from the inside. And several things are happening when calcium enters to the NMDA receptor. One is that it turns out there were already AMPA receptors that have been built, that were sitting there, but they get inserted into the membrane right away. And so that means that after the tetanus, because there are more AMPA receptors, now when we go back to that slow, steady stimulation, the EPSB is larger. And it's going to stay larger as long as those AMPA receptors are still there because there's a bigger postsynaptic receptor array. And when that was first established, people said, in LTP, the synapse gets stronger because of a change in the postsynaptic target. It's got more AMPA receptors there. And people were very keen to ask, well, and is that all there is to it? Oh, I should say, originally the question is, well, okay, LTP is there, it's real. Is that a change? Is it causing a change in the postsynaptic cell or the presynaptic cell? And the first answer came out quite clearly, the postsynaptic cell was being changed. Though eventually it turned out, well, the presynaptic cell is being changed too. Because another consequence of the activation of the NMDA receptor are complex changes that eventually cause the postsynaptic cell to release some sort of retrograde signal. It's called a retrograde signal because it's going in the opposite direction in which the information normally goes through synapses. And what the retrograde signal, no one knows exactly what it is. There's some candidates out there. But whatever it is, it tells the presynaptic cell to release more neurotransmitter next time. So the changes that happen after LTP are twofold. Number one, the postsynaptic cell is more sensitive because it has more AMPA receptors there. And number two, the presynaptic cell is releasing more neurotransmitter. And these changes can last for hours or weeks. How's that? You can see that the NMDA receptor, this pairing of NMDA receptors and AMPA receptors, it's perfect for having a synapse behave in a Hebbian fashion. Only when the postsynaptic cell is already close to firing will the magnesium block be lifted, and therefore the NMDA receptor can do its deed.
So when Mark's potentiation, does that always occur in glutamatergic neuron?
It's a great question. No. In fact, there are some places, even in the hippocampus, where you can get long-term potentiation, and the neurotransmitters are opioids. Right? Yeah. So there are, but far and away, the LTP, glutamate-mediated LTP to the NMDA receptors, far and away the most studied. And frankly, I actually don't know much about the other, but I know they exist.
The magnesium that netted because the cell has become too toxic.
Exactly. So the magnesium block is lifted because the cell isn't as negative as it used to be.
How can we all drive like sodium or
Why is calcium attractive? Because it's just to do with the equilibrium potential of magnesium versus the equilibrium potential of calcium, right? Without going into any more detail. So the cell is still negative inside, but it's not negative enough to counteract magnesium's concentration gradient.
and that's they still do it.
Yep. So it's still getting depolarized, and calcium still wants to come in. And what happens after that is more complex and really hasn't anything to do with development, but has to do with synaptic plasticity, and you'll take another course for that. So these are the so-called short-term changes that happen in LTP. And I think I'm going to skip over this, but here you can look at this, that in addition to that, there can also be long-term changes in the strength of the synapse, all of which was triggered by LTP. The idea is that, of course, there are not tetanuses normally in the nervous system. The idea is that if instead of having, say, one input fire repeatedly to make the postsynaptic cell fire, you could also imagine that there's a convergence of inputs. And if that input from several different neurons, if they happen to arrive at the same time, they could depolarize the postsynaptic cell enough to activate the NMDA receptor cascade. How's that? And again, synapses that successfully cause the postsynaptic cell to fire would get stronger. It's a situation where, again, the American way, the rich get richer and the poor get poorer. Synapses that are successful get more successful, and synapses that aren't successful become less successful and may even wither away entirely. I think we have time for another Kahoot.